tonight we're discussing male infertility, today's insights, tomorrow's treatments. And a huge thank you again to the Scottish Government for their sponsorship of this event and to the Society for Reproduction and Fertility for all their support on the ground. I'm Sarah Norcross and I'm going to be chairing the event, and I am really excited to be here this evening in this beautiful lecture theatre. We'll now invite the first speaker. We have Kevin McEleny. It seems very strange to introduce you. Chair of the British Fertility Society. He's a consultant andrologist and clinical lead at Newcastle Fertility Centre and consultant urologist at the Freeman Hospital in Newcastle and the Queen Elizabeth Hospital in Gateshead.
These are my disclosures. I'm speaking in a personal capacity this evening, though, although I've got some honoraria in the past. Really, my talk is really about why is this an important topic. We've heard some of the information from Sarah just a minute ago about it, but I think it's a really important topic. Firstly, because it's very common. It is a common reason for couples attending clinics with problems to do with their fertility. There's a male issue involved. It's the only issue in maybe 30% of cases, and if you factor in situations where there is both a male and a female issue, it's there almost half the time. According to some research, it does appear that sperm quality could be declining worldwide. Some of the early data came from Scandinavia, from Skakebak and others, but more recently there's been this paper which showed across what they describe as northern countries, which also include Australia, somewhat bizarrely, Europe, North America, Australia, there's been a significant decline in sperm concentration over a 40-year period of 50 to 60%. A follow-up study by the same authors looking at southern countries, as described, South Asia, Africa, Latin America, showed the same thing. Not everybody believes this, but there is some suggestion that sperm quality could be declining over time, and if so, that could be a problem. It's also important because we now know that it's associated with other health issues. Now, we do know that there's certain genetic problems that can be linked to fertility in men. Some of these kind of other impacts on male health and their risks for particular problems. Hypogonadism, a low testosterone, is frequently seen in our patient population. This can significantly affect male health and may also, according to work done here in Edinburgh, in fact, be becoming more common also. Obesity, again, one of the commonest health problems of our modern age, is linked to male infertility, as it is linked to diabetes too. So you can see a few kind of really common themes coming through this in terms of the impact of fertility in male health as interlink. Now, we've known for some time that testes cancer is more common in men with fertility problems, with sperm quality issues, but we now know prostate cancer, colorectal cancer are also more common. The reasons for this are really speculative. We don't really know why, although my buddy Joris Veltman may have a few ideas on this, I suspect, from genes discovery that we've carried out. Cardiovascular disease is too more common, and Eisenberg and others have shown this work. We don't really know why, but it could be, as has been described, this decline, or this possible decline in sperm quality is the canary in the mine warning us in society that something's going wrong with male health. I think it's quite interesting is that they're kind of, I suppose, are they cultural? There's certainly educational differences between men and women and how they approach fertility. In general terms, men don't have the same understanding of how their bodies work. Most men don't, as women do. So if you tell them that, for example, there's no sperm in their ejaculate, it'll come as a tremendous shock to them. A lot of our patients are very anxious, and many of these patients will do well with counselling, but not all of them will. Men may be more inclined to internalise anxiety, to keep it bottled up, and when it's not clear whether traditional counselling is in fact the best way to support our male patients, they may need something else instead. There's an unhelpful concept called traditional masculine identity, where a man is supposed to be strong and absorbing of pressure and silent and fertile and potent. These things are all wrapped up together. It's not a terribly helpful thing, but it is what we have. And when you start to chip away at this by telling a man that he's, for example, infertile, you do it does have an impact on how he sees himself. Now, Sarah's mentioned that it's less of a taboo perhaps than it used to be, and that's great. I've definitely seen changes in this, dramatic changes over the last 20 years. But elsewhere in the world, it is very much a taboo, and we're looking at situations where women who are involuntarily childless can in fact be subject to violence as a consequence perhaps of their male partners being infertile themselves. It's a really big deal. This is information that came back from some years ago from the European Association of Urology. They were saying that in 30 to 40% of cases overall, there was no clear cause of the reason for the man having a sperm quality issue. And I find that data a little bit hard to believe, because when I've got the patient sat in front of me, it's very difficult to say that this is the specific reason why they have the problem. If you do something to them and they get back to normal, well, maybe you found it. But I think it's much more complicated than that. And I think it is unusual to find a specific cause in a male patient. And there is certainly a lack of high quality clinical evidence on interventions. Up and down the country, male patients are being charged for treatments that there isn't a strong evidence base behind them. And this is something of concern. So we need better studies to confirm the role of such interventions. I think assessment is important, because if you can assess the patients well, appropriately, you might find what the reason is. And so much of what we do is in terms of the patients wanting to know what is wrong, why are they in that situation. We're not good at it. But if you do find a problem, for example, you might enable them to have a less invasive way to form a family. We would much rather our patients conceive naturally than require assisted conception. And along the way, as mentioned earlier, we may find other medical problems. So, taking the history of the male patient, you ask about the fertility history, have they had children before, have they been investigated with their partner. You're interested in general medical history, because general medical things are relevant, but particularly history of any testicular and genital problems, significant injuries, infections and operations. Poor health issues, chronic health problems can be relevant, as can a surgical history, particularly of things that can affect sexual functioning in men, pelvic injuries, pelvic surgery. A drug history is important, but remember to ask about over-the-counter preparations, which can be relevant, or indeed recreational drugs, particularly anabolic steroids, whose use is at kind of epidemic levels in younger men. Developmental history can suggest if there's a delay in going through puberty, maybe they've got a genetic issue or an endocrinopathy. A family history of cystic fibrosis might be relevant. Sexual history is important. We see increasing numbers of men coming through really with ejaculatory problems, most of whom, in my practice at least, will be type 2 diabetes, although it's most frequently described in men with spinal cord injuries. And erectile dysfunction, again, is something that can be fixed in men and can enable them to form families without, perhaps in many cases, too much intervention. I won't talk about environment and lifestyle factors, as you've got a world expert sat who's I'm sure will be talking about them a bit later on. When you look at the male patients, you look at the body habitus. Could there be a genetic problem? Could there be an endocrinopathy? Obesity can have an endocrine disorder linked to it. You look at these kind of things, but the most important thing is the examination of the genitals. The testicles should be at a minimum size. If they're smaller than that, if they're soft on palpation, it does indicate a production type problem. And you see Professor Anderson sat there, a keen farmer who, of course, knows this only too well from his livestock. One thing to bear in mind is that testes cancers are more common in this population of men, and you do find them occasionally. So we do tell our patients to self-examine, and if you do find a lump, get an ultrasound scan. The pipes that carry the sperm can be missing, particularly in men who've got cystic fibrosis gene mutation problems, which is obviously one of the commonest genetic problems in Northern European heritage people. They may be absent. The caps, the testicles, the epididymides, you can see the diagram, can be swollen. That can indicate also a blockage. Varicoceles are dilated veins draining the testicles. They're more commonly seen in infertile men. Treating them does seem to improve sperm quality in most studies in which they've been carried out. The impact on pregnancy is perhaps less certain, but there are certainly studies that say that treating varicoceles makes a difference. Internationally, this is in guidance so that men who are infertile, who have reduced sperm quality and who have a clinical varicocele, one that you can detect on examination, as our practitioners should be doing, those men should perhaps be treated, and this is coming into the new NICE guidance. So male patients may have some options available that they didn't have perhaps in the past. So semen analysis is the most important single test, and we're really dependent on our labs providing a good service, a good quality assurance-based practice. And if the quality is low, we do hormonal and genetic studies, looking for things that might explain or be associated with it. Occasionally we do imaging tests. Joris will be talking about genetic problems, I suspect, in detail, but a significant proportion of men, they say, have genetic problems. The actual proportion is relatively low in those in whom you can detect. So looking at men with non-obstructive azoospermia, a production type problem, maybe 10 to 15% of them will have a genetic problem that you can identify. the majority of which is going to be Klinefelter syndrome. The Y chromosome contains the sperm-making genes, and here in Edinburgh, in fact, back in the 90s, it was identified that certain patterns of fertility problems were linked to these microdeletions. Cystic fibrosis testing is interesting. They test for maybe 20 to 30 genes, but there's over 2,000 different mutations that are available, and they can be ethnically based. Sperm retrieval is an option for those who don't have utilisable sperm in their semen or a treatable cause of their infertility. Most classically done in men who have no sperm at all. If it's obstructive azoospermia, there's a range of procedures that you can do. If it's not obstructive, now microtesting is recommended, a more invasive operation to try and get sperm. It's said to have a higher recovery rate. So in terms of my own personal views for the future, I think we need better diagnostics to identify the causes of male factor infertility and to predict success with treatment. We'd like clarification of the role of any medical or other adjuvant treatments. They're being done, but do they work? I think that in vitro generated gametes will be transformative, and I think they're just about to, I'm just about to finish. They'll be here in maybe 10 years or so. I think that will be a really big development. But for the moment, although it's less of an issue in Scotland, of course, the thing that would help most men the quickest would be full implementation of the NICE recommendations in NHS patients for fertility treatment.
So our next speaker is Professor Joris Veltman, Director of the Institute of Genetics and Cancer at the University of Edinburgh. He's an executive board member at the Human Genome Organisation.
So I'm going to talk to you about genetics. And as geneticists, we used to work with large families where disease occurs in a family, because we could then look at markers that associate with disease and in this way find the genetic cause of disease. However, when you think about genetic diseases, many of them actually are not occurring in big families, and actually the disease itself has an effect on fitness. And this, of course, is very well explained if you think about infertility. So a man with infertility is born to fertile parents, right? The parents were fertile because else he would never have been there, unless, of course, he was born through assisted reproductive technologies, which nowadays is actually something we can talk about. But this means that we have very small families, and that's something that we've been looking at for a long time. And what we can now do when we think about genetic cause of infertility is look at the entire genome. And this genome, of course, is enormously large, 6 billion letters, and just a single letter change can result in a severe disease. However, I'm a technologist, and technology changes everything in life, from mobile phones to genome sequencing. When I was a student, you could look at, let's say, one gene at a time. Now our students can sequence their whole genome in a matter of hours and get it into the computer to do fascinating work. And this has changed medicine, because if you look at some of the papers from recently, you see that genome sequencing, genetics, is now becoming a primary part of medical services. Patients with rare disease, patients with cancer will get a genome sequenced in order to determine what the best treatment is. And this is really critically important, and this is really beneficial for healthcare. So where are we in the male infertility field? Well, Kevin already mentioned it, Klinefelter syndrome and extra chromosome X, microdeletions on the Y chromosome, they have been known for a long time to cause non-obstructive forms of azoospermia, but they explain only 10–20% of cases. What we have to look at for the large majority of cases is indeed look at these individual genes where a mutation, a single letter change in your DNA in one of these genes can cause disease. And what you see here on this slide is a number of genes which we've associated to infertility, but actually that number, while it looks impressive, is still very low, and actually that means that we cannot find the cause in the majority of patients. So as Kevin mentioned, 5–10% will have chromosomal abnormalities, 2–6% will have Y chromosome microdeletions, but the large majority, these mutations in genes, they are basically underexplored and also not used in diagnostics. So that's a big problem. What's the consequence of this? Already mentioned, if we don't know what the causes of your infertility is, we cannot treat you. You cannot treat your individual cause of your disease. So you have no personalized medicine. And that means we have no idea whether the assisted reproductive technology will work. We have no idea whether you're passing on infertility to your children if ART is used. We have no idea whether indeed this man may perhaps also have a longer-term risk of cardiovascular disease, of cancer. And we, of course, will not develop new treatment options. So we really need to know more about why are men infertile. And this field, I think perhaps because of the success of IVF and ICSI, is very much driven to let's get this man pregnant, but not understanding the cause. As Kevin mentioned, it's really important. So in international consortia, what we can now do is use this genome sequencing approach to sequence large numbers of patients and then share data together. And in this way, we and others have really, changed, let's say, the number of genes that we can now associate with severe male infertility. So that's the good news. We can use these approaches to further our understanding and find all these different individual genes. And then go to the patients with mutations in these genes, look at the success of their STISA, look at comorbidities, look at all these other things. So that's where we can learn a lot. And I think this is just to drive that home, because not always clinicians, they think, well, perhaps genetics is not that useful. Well, actually it is. If you look at the well-known microdeletions on the Y chromosome, depending on how big the deletions in the Y chromosome, you either can predict that the success of TISA will be 50% or it will be 0%. Well, if you know that before you start, it means you can actually tell this man, don't go for this. Perhaps you need to think of other ways to go. And if you then look at individual gene mutations, for example, the one here on the right, sometimes genetics is actually very good news. So if you have a mutation in the CFAP47 gene, which is a gene on the X chromosome, actually we know that all men with mutations in this gene actually are successful in becoming a parent with assisted reproductive technologies. And because this gene is on the X chromosome and it's in your man, as everybody who does genetics, it means actually you will not pass that on to your son, because he gets his X chromosome from his mother and the Y chromosome from father. So that means it will not be passed on to your child. So genetics is important. The Fanconi M gene is, for example, an example where cancer is involved. So there is a lot happening in the field. A number of colleagues around the world have started to implement these genetic tests now into routine diagnostics, not yet in the UK, but colleagues see that when we use these kind of tests, we get diagnostic result which is comparable or higher than testing actually the standard karyotype or the microdeletion on the Y chromosome. One of these colleagues is in the audience, Margot Weerwald there. I'll have a picture of you later, Margot, so don't worry. So that's great news. So we're starting to use this in diagnostics around the world. And the important thing for researchers is that we can then also use it to find novel genes that actually can, again, increase the diagnostic yield, because we need to do this in a large setting. So bringing this back to these plots where we basically, in a graphical way, represent a family. If you think about this and you think about genetics, how can you look at this? And one of the things you would do if you have an infertile man, you would actually look at the DNA of this man, but compare it to the DNA of his parents. And that's something that we and others have pioneered for other diseases, but we've now started to do this also for infertility. And together with my colleague Kevin and others, we have collected the world's largest cohort of infertile men with their parents. And if you look at that, no surprise, you will find that a man inherits half his genome from his father and the other half from his mother. So that's not a surprise. However, very importantly, if you look at this genome of this man, you will find a number of mutations in the DNA that you cannot find back in the DNA of his parents. And that is because during production of sperm and eggs, actually new mutations arise that are passed on and become part of the germline of the next generation. And these mutations are critically important. So we have pioneered this for intellectual disability, for mental retardation, but more recently we've pioneered this also for male infertility. And this provides us with a new insight into dominant causes of infertility, which again are very important to know, because it means that you actually have a 50% of passing these on to your offspring if you are using assisted reproduction. So that's a field we're looking at. We're finding new biological pathways that are affected, which again are critically to further investigate, and that points us to novel, let's say, treatment options, as already mentioned. So this is work that is ongoing. Here are the pictures. So I've recently moved from Newcastle to Edinburgh, and now working with two amazing colleagues here in the room, Margot and Alex, and we're hiring, by the way, if anybody is a PhD student, wants to do a PhD or a postdoc with us. We want to further understand the genetic cause of infertility. It's really an understudied field, but definitely also demonstrate the clinical value of genetics, implement this into the clinic, because why is this not done for infertility, whereas it is done for rare disease, for cancer? The UK is leading in this. I have to be honest, actually, England is leading in this. Scotland is way behind. right? So we need to do something in genetics in Scotland for rare disease and cancer in general, but definitely for infertility for the entire UK. So we need to raise awareness, and PET is a fantastic organization to do so. So I'll stop here. This is work from a lot of colleagues from around the world. I think it is fantastic that we have an international consortium to work on.
So we have Professor Rod Mitchell, and he is Professor of Developmental Endocrinology here at the University of Edinburgh. He's Research Group Leader at the Centre for Reproductive Health, and he is Lead for Male Fertility Preservation at Edinburgh Fertility Preservation, and Principal Investigator of a pioneering fertility restoration project funded by the charity Children with Cancer UK.
So what I'm going to talk about is, I've called it a fertile future, and I hope you'll probably see why by the time we get to the end. But this is about preserving fertility in boys with cancer. So I want to just start by telling you about a boy, a 5-year-old boy, who came to Children's Hospital because he'd become a bit wobbly, become a bit unsteady on his feet, and he'd had increasing headaches over the last 4 weeks. And that's a very worrying thing for us when a boy comes in with that kind of story. And so he had a CT scan very quickly, and it discovered that he had a mass in his brain. And he actually had brain cancer. He had a type of brain cancer called medulloblastoma. And we knew that this boy was going to get treatment, and he's going to get quite heavy treatment. So he was going to get a combination of surgery, radiotherapy, and chemotherapy. And of course, what we know is that these treatments, while they can treat cancer, can also cause problems. And one of the main problems that particularly chemotherapy and radiotherapy can cause is problems with fertility, and it can damage the testicles directly. So if you imagine this young boy and you try to think, what can we do to help him from a fertility point of view? Well, of course, if he was a bit older, if he was an adult, then we could obviously collect sperm before he had his treatment. And that's something that's quite well established for fertility preservation in pubertal and adult males. But because he's still a child, he's not making sperm, he's too young to make sperm. So we have to have another way of how we can preserve his fertility. And the way we would do that is to try and preserve the stem cells, the cells that will make sperm eventually but aren't currently doing it. And these cells are called spermatogonial stem cells. And so the way we would do this is that this boy, before he had his treatment, would have a little piece of his testicle taken surgically. It would be frozen to preserve those spermatogonial stem cells, and then with the hope that we could use that tissue to restore his fertility in the future. So we're preserving potential for fertility, not guaranteed fertility. Now, I'm going to show you a picture of the surgery itself. So if you are of a squeamish nature, or you don't particularly like looking at testicles, then you can look away just now. But this is essentially what it looks like. This is the surgery that we would do. The testicles taken out through the scrotum, and a small incision is made, and a little wedge is taken out of the testicle. It's sewn back up, and it's put back in. So you can see it's a relatively small operation. It doesn't require taking a lot of tissue, and it does leave that testicle, the remaining testicle, in place just in case this individual's fertility is retained. So we've been offering this now for quite some time in Edinburgh, and we have a criteria of who we would be able to offer this to, and you can see it listed here. But the key things are that these individuals ideally need to have normal testicles before we take the tissue, obviously, to have potential for fertility. And we offer this to patients who are at the highest risk of infertility as a result of their treatment. And we've been doing this in Edinburgh for some time. We've been collaborating with colleagues in Oxford to provide a service across the UK. And we've also developed now a consortium, an international consortium, of all the centres that are offering this for patients. And we know that the number of boys that are having this done is increasing quite rapidly, so that more than 3,100 boys, the last time we did the survey, had testicular tissue taken. So David, who was 5 when he had his testicular tissue cryopreserved before he started his treatment for medulloblastoma, is cured of his condition, and he comes back to the clinic at the age of 19 because he's concerned about his fertility. So the question then is, when he comes back, how do we actually assess his fertility? What do we do about it? And we heard in the last talk, in Kevin's talk, about how we would do that. And the key thing we would want to do for this young man is to get a semen sample. That's the best way of determining whether or not he's going to have fertility. So he has his semen sample, he produces his sample, and they have a look at the sample. And unfortunately, he's got no sperm in his ejaculate. So at the moment, this young man is infertile. But of course, we did store some tissue from him. So the question is, what can we do with the tissue that we have in the freezer? And the main way in which we are thinking about doing this at the moment is to take that tissue out of the freezer, to thaw it, and then to transplant it back with the hope that it will do what it's supposed to do and start to make some sperm. So there's two ways in which you can transplant it, and you can see here on this diagram how that works. The top option is to take the tissue, thaw it, extract those stem cells, the cells that know how to make sperm but don't yet, and inject them back into the testicle to try and restore fertility normally and have natural fertility. That's one option for transplant. The second option, which is the bottom option, is to take the tissue out of the freezer and thaw it, but instead of isolating the cells, just put the tissue piece back, hope that that tissue piece will start to make sperm, and then you can extract those sperm and use it for artificial reproduction, inject it into an egg and generate an embryo. And these are the two approaches that at the moment are being considered for clinical application. So I'm only going to talk about the tissue transplant for now. We can talk about the stem cell transplant later, but this is the one that we probably find most promising. And the reason we find it promising is because 6 years ago, when I last spoke at PET, I talked about this, and this is a transplantation study. This was an experimental study that was done in rhesus monkeys, and they took a prepubertal rhesus monkey, they collected a piece of his testicle, they then treated that monkey with sterilizing treatment, and they transplanted those bits of tissue back to the monkey. So essentially what we're talking about doing in clinical practice. And when they retransplanted the tissue and took that tissue back out after around about 9 months, that tissue had started to make sperm. So we knew from that it worked. And they took those sperm. This is a group in the States that did these particular experiments. This is Kyle Orwig's lab. They took those sperm, they injected them into an egg, and they generated an embryo. They generated a pregnancy of a rhesus monkey. And they had a live offspring. And this is Grady. Grady is so called because she's graft-derived baby, which is where she gets her name from. And 6 years ago, I stood, not here, about a mile away, and I told you that this is something that in 5 to 10 years might become possible for humans. And of course, scientists, clinicians always say that, and often it doesn't happen. However, we got together as our OrchidNet consortium, and we actually talked about whether or not the time is right to do this for humans, to transplant tissue back into patients. And we came together with essentially a description of how we think this should happen if we were going to do it for patients. And this was through our OrchidNet International consortium. And then Ellen Goossens, one of the team from that consortium from Belgium, that's Ellen there, you can see her on the left-hand side. Ellen, right-hand side. Ellen did indeed do the first testicular tissue transplantation from a boy who was now an adult who had had testicular tissue stored. And she did that transplant on the 10th of January, 2025. And of course, that tissue has to stay in place for a period of time to start generating sperm, so they decided that would be around about a year to keep that tissue in place. And then 6 days ago, and you are probably amongst some of the first people to hear this, to know about this, Ellen published her paper, which is still as a preprint, it's not been peer-reviewed yet, but to show that complete spermatogenesis, sperm production, could happen in those testicular tissue grafts. And that's just one image from the paper, but please go and read the paper, have a look. It's obviously, I'm excited about it. I hope you guys are all excited about it. That's a picture of the paper of the tissue that had been grafted and retrieved after a year. And there you can see in panel B there, that's one of the sperm that they generated from their testicular tissue transplants. So it works. We are less than 10 years since I told you it would be within 10 years. What we don't have yet is a baby, and hopefully that will be the next step with this. But as well as the team in Belgium and one or two other centres around the world, we are approved now to do testicular tissue transplants for our patients, the boys that we've taken tissue from over the last 10 to 15 years. And so we're looking for patients who meet the criteria. It's fairly straightforward. They need to be adult, obviously. They need to have had testicular tissue stored, or there's nothing to put back. And they need to be now recognised as being infertile. And we're hoping to do some of these ourselves in the very near future. So I said it was a fertile future. I honestly think this is a fertile future. I think this is a great step forward for the field. I should just acknowledge members of the team that are involved in the work that we do here in Edinburgh, members of the research group, but particularly in the middle there, the Edinburgh Fertility Preservation Group. You can see actually it's a lot of people that are required to run a fertility preservation programme. Several of them are dotted around the audience as well, so thanks to them as well. Thanks to all the team that are involved in this, and I can honestly say it can't be more of a pleasure to be involved in this particular area just now. It's so exciting, so thank you.
So our next and final speaker is Sarah Martins de Silva, Professor of Reproduction and Fertility Medicine at the University of Dundee and Clinical Lead for Fertility Services at NHS Tayside. She's a consultant obstetrician and gynaecologist at Ninewells Hospital's Assisted Conception Unit.
I think we're all very aware that infertility is a really big deal, a really common problem, and probably quite an unseen problem that carries quite a lot of unseen health burden in terms of mental health as well as relationship strains and so on. We've already heard from Kevin that male factor accounts for nearly half of all cases. And we're all very acutely aware that apart from very exceptional kind of niche cases, there really are no treatments to directly treat infertile men. So couples have very limited options. They tend to go down a pathway of IVF-ICSI. There's a huge unmet clinical need for the development of new treatment options for male infertility. So this kind of slide is just a cartoon of what our sort of overarching kind of research picture is. If we can understand how sperm work, then we can understand why they don't work. And if we understand why they don't work, we can create new treatment options to try and correct that dysfunction, and therefore treatments to treat men directly to allow them to conceive without lots of medical intervention. But somewhere along the way, we also worked out that actually if we could perturb sperm function, actually we might come up with some other options that aren't hormonal-based contraceptives. So I've got some key concepts for you here. We all know that spermatogenesis occurs in the testes. It's a continuum that's driven by hormones, and it takes probably about 70 days to build a sperm from start to finish. That happens in the testicular tubules. The testes then leads through the tubule structure into the epididymis, and there's a further 10 days to 2 weeks or so where the sperm is matured in the epididymis. And at that point, the sperm cells are quite quiescent, and it's really only on ejaculation, probably in a response to change in pH, that sperm start to be able to move and swim. But when they're first ejaculated, they can't fertilise an egg. They need to then go through a series of other biological and molecular changes, which is kind of termed capacitation. This is where there's changes in the membrane, the protein expression, the lipid expression within the membrane, and more. And so that the sperm can then change how it swims to different forms of swimming. We call one of them hyperactivated motility. There's ion channel modulation, and also the ability then to acrosome react and release the acrosin from the head area. And there's also some other sort of sophisticated interactions that need to happen. The sperm needs to bind to the zona pellusa, which is the jelly-like egg coat. It needs to fuse with the egg membrane itself, and there's specific named membrane proteins there from the egg and the sperm. And it needs to carry into the egg a protein message called phospholipase E to activate the egg. So there's a huge amount that we know, but there's also a huge amount that we don't know. And actually there's quite substantial challenges and difficulties working with sperm cells in a research setting. They're very small, they move around, they virtually have no cytoplasm, so they behave very differently to most other cells, and therefore all of the assumptions that you might make about cell signaling and so on doesn't really necessarily hold true for sperm. They're also terminally differentiated cells, so you can't culture them, and generally speaking, if you create a knockout mouse model, then they are infertile and therefore a fairly self-limiting experiment. But what do we know? Well, what we do know is that actually because of this very limited cytoplasm, sperm don't build and transcribe genes and build proteins and so on. They rely on the interaction in the environment that's around them. And we know that particularly intracellular calcium and changes up or down in intracellular calcium is utterly critical for how sperm swim, as well as other function required to fertilize an egg. We also know that an alkaline pH, so a low level of hydrogen ions, is important. Both intracellular calcium ions and hydrogen ions are primarily determined by ion channels or pores within the membrane, as I say, to import or export these ions. Although there are some calcium stores within the cell as well. Lastly, there's a very hyperpolarized membrane. It's maintained by a potassium channel, outwardly going potassium channel, to maximize cat-spur function. So in terms of function for sperm, there are a triad of ion channels: cat-spur, which is a calcium channel, K-spur, potassium channel, HV1, a hydrogen channel, and these are absolutely fundamental for sperm function. How do we know that? Because we've looked at patients and we see a high level of ion channel dysfunction within cohorts of patients that go through IVF ICSI and end up with low fertilization or no fertilization. So K-spur, probably about 10 to 15% of patients with low fertilization at IVF, and cat-spur defects probably account for about 1% of male infertility populations with fertilization issues. So we've tried to leverage this. We know that sperm motility is important both for natural conception but also for IVF success. So it's a very attractive therapeutic target. And we've gone through a series of collaborations with the Drug Discovery Unit, part of the University of Dundee, to try and work towards kind of screening libraries to try and find candidate therapeutic agents for male infertility. Now, this pipeline is going. It's a slow, long, drawn-out process, and unlike Rod, I'm not going to promise you that we're necessarily going to arrive at an answer within the next 5 to 10 years, but maybe. In the meantime, what I want to do is circle back, because I think Kevin's alluded to this, and maybe Rod has too. We have a way of determining male fertility prognosis, which is diagnostic semen analysis. It's a quantitative assessment which involves producing a sperm sample by ejaculation and highly qualified laboratory technicians counting sperm, associating the movement and motility, and looking at morphology. But it's a very quantitative, paper-based algorithmic assessment that hasn't really changed in over half a century. And the only thing it tells you really is whether you're likely to have a fertility problem or not, unless there's no sperm at all. So the big question I feel that needs to be asked today is there a better way to assess male fertility? And I think we would all agree, yes, there must be, right? So what we are currently embarking on is a Gates-funded study trying to determine what the thresholds for male infertility might look like. It's a clinical study which started last year, but we've started actively recruiting patients since January of this year. And it's a collaboration between the University of Munster, Dundee and Birmingham. And what we're trying to do is encourage 1,000 fertile men and 1,000 infertile men with unexplained or mild male factor infertility to complete a health questionnaire, submit a sample for diagnostic semen analysis, so the current gold standard of what we're assessing within a clinic, but then a raft of highly standardised sperm function assays and tests. And a big shout out to Hannah and Annie that are in the audience here who are doing a lot of this work. So we've chosen to try and evaluate key parts of that sperm function. We're looking at capacitation, hyperactivation, and the acrosome reaction. We're looking at that in a lot of different ways. There's some pictures here. You can see one of the really funky things is this flagella or fast analysis, where we're looking at patterns of the flagella beat in response to various agonists, and also then looking at the acrosome reaction in response to different stimuli. This is going to give us some really exciting but hugely complex data reads out. We're going to look at bioimaging and microscopy. We're going to do multivariate analysis and use sort of really kind of sophisticated AI and machine learning to try and look at all of these different variables to see if we can see the difference between fertile and infertile men in a much more dynamic and informative way. Clearly, this is a high-risk, high-reward project. We might find no difference at all, and that would be possibly quite disappointing, although I think Mr Gates has got enough money to possibly not mind. On the other hand, our real vision here is to try and improve the understanding and the diagnosis for these men who are struggling to conceive, who often face no answers in particular. But also, this will also inform drug discovery pipelines, both for male fertility but also non-hormonal contraception. So to finish up, I think we all agree male reproduction is an important area. There's a lot of unmet need within this niche, and we do really just need to understand the fundamentals better. I still, to this day, cannot understand how we can put a man on the moon, and yet we do not know how does a sperm swim, find and fertilise an egg. We want to improve the diagnostics for men. We want to improve treatment options for men rather than directing all of their partners down an IVF-ICSI pathway. And I think even having information from a diagnosis is an empowering thing for the men affected. So clearly changing the landscape in this field of research will have an important impact not only for Scotland, not only for the UK, but across the planet.
Joris, thank you very much for your insights. I wonder whether or not you could speculate how many male children of people that have been conceived with ICSI will have a sperm count pretty much identical to their fathers.
Yeah, that's a good point.
So you want to use the mic?
So, great question. Actually, a colleague Tournaille published some work on that a number of years ago, where they looked at boys conceived through ICSI and looked at their sperm counts. And to be honest, it wasn't completely clear, but there was a fraction of the boys who clearly had infertility. Not all of them, and that's also what you would expect as a geneticist, because of course, you know, if it's a dominant cause, you would expect it in about half of them. If there is some recessive causes of infertility, you would actually not immediately expect it to be, let's say, coming back. So I think we don't know. I think this is indeed something that Sarah alluded to. We should know much more about it. You know, all men should know their sperm count, right? That would be lovely if we would regularly do that, because that would help us enormously. So, for example, if you think about the UK Biobank, it would be great if we would, you know, start, you know, looking at a cohort and recognizing that, because I think that's a big problem. We don't have that. But from the genetics point of view, I can only say, you know, there is absolutely going to be lots of boys who have a problem. We are passing on infertility. You know, this is just, you know, you're basically, you know, that's can't be any other way.
I've got a question for Rod. So I noticed that with the spermatogonial stem cell transplants, you get natural fertility, but with the tissue, it said they needed ICSI. And also in the picture that you showed for that recent paper, the tissue had been removed. So is it kind of a one and done situation, and how many sperm you get, that's you done forever? And what's the difference between the tissue versus the sperm cell process?
Okay. So they are different for that respect. Tissue transplants, you're always going to have to take the tissue out, extract the sperm, and use those for ICSI. It's never going to give you natural fertility. And once you've taken the transplants out, whatever sperm you get is what you've got. You're not going to get any more unless you've got more tissue to transplant back. The stem cell transplant is basically transplanting those spermatogonial stem cells into the tubules of the testis. And we know from the animal studies that those spermatogonial stem cells go back to where they were meant to be. They find their way back somehow, and they start to make sperm. And those sperm can be ejaculated just like normal. And so that can then give you natural fertility. The problem with the spermatogonial stem cell is twofold. Firstly, that it's a bit more technically challenging to do than the tissue transplant. So that's practical, logistical issue. The second is, and I didn't say this, but it has been done, again, around about a year ago, and again by another member of this international consortium in the States. And we're waiting to find out if he's going to get sperm production in the testis and in his ejaculate. The problem will be that even if he does, we won't know for 100% sure that he didn't recover his own natural fertility in that testis. We won't know it will come from a stem cell that we transplanted back. So that's going to be a challenge going forward, because we would probably need a lot of successful transplants before we knew on the law of probability that these did come from the transplanted cells.
I think one of the slides you showed, Joris, was something like 10 to 20% of the men studied were found to have a genetic cause. Do you think that's actually the limits of modern genetics, that sort of number, or are there going to be more advances in the level of the depth at which we can analyse things that's going to put that number up substantially?
Yeah, thanks, Richard. Great question. I actually find this always difficult. You see these reviews where people say, 20% or 30% of infertility is genetic. And I think, how did they know that? Because nobody's looked at the genome and gone through all of that. So I think there is a problem in general that we don't know how much is genetic. And when we, for example, say it's hormonal, well, actually, hormones are also, again, there is genetics there. So as a geneticist, I think everything is genetic. But I think clearly, at this moment, we don't know, I think I would not more than, let's say, 10 or 20% of the genes that can cause infertility. So basically, we're missing the majority. So even if we can, now sequence the entire genome for about 100 pounds, we don't know how to interpret the variants. And I think we are, this field is way behind any other medical field. It is like, like this lecture theater. We live decades behind modern, let's say, medicine, where, a patient with cancer will get a genome sequenced. There is, personalized treatments that are now being developed. And in our field, we do a karyotype. So yeah.
Is there any hope for people who have extra chromosomes, for them to have sperm that can maybe in the future be used to help them have children? And the second thing is about trying to use somatic cells. I know this is science fiction. Well, it's not science fiction, it's just beginning. Trying to use somatic cells to derive eventually sperm cells.
So I think for the extra chromosomes, then there's definitely animal studies that would suggest that there are ways in which you might be able to get rid of extra chromosomes to then be able to generate gametes from that. So there are studies that lead along those lines. There's nothing yet towards human or clinical application in that area yet. For the somatic cells, that is a really rapidly progressing area. And certainly, again, the animal studies which would show that you can take skin cells, you can reprogram them into germ cells, you can take them through to gametes, and you can generate offspring from those. So that whole proof of principle is there in animals. People are working very much towards doing that now for human application. Having said that, ethics, safety, future generations, there's loads of questions about that, but it is rapidly progressing.
So with the male infertility research, has there any crossovers between male contraceptives work that's been brought to light from all the research, and can you share some of that?
So 100% yes. So what we did was, first of all, we created a robotic-generated high-throughput screening. So for drug discovery, we just screen loads of little molecules and druggable molecules that you think could, would be the basis of a drug that will get to where it needs to get to in the body, so it needs to be well absorbed, etc. So we originally screened looking at calcium responses, and we found a whole kind of raft of different target drugs, and it's really interesting if you pull some of them apart. So one of the ones I put on the slide was a drug called traquinzin, which is in the same sort, it's a phosphodiesterase inhibitor, it's the same sort of family as Viagra and theophylline and so on. So some of these from that same family don't improve sperm, but theophylline does seem to. And theophylline you may recognise is an asthma treatment by choice at the moment. On the other hand, then what we did was kind of moved on to a more sophisticated way of screening drug responses, looking at how sperm moved. And that was a project funded by the Gates Foundation, and we were trying to develop a contraceptive. So we were specifically looking for significant depression of sperm swimming. But of course, within a drug discovery pipeline, you'll always have those that do what you don't want them to do. So we've got a whole raft of different data that shows where sperm motility has been uplifted. The real criticism or the real kind of blockage with doing that type of drug screening project where you're looking at just what the phenotype is, in other words, what it does to the cell, making it swim better, making it swim worse, is you're not aware of what the target is. So we're now at a point where we've got probably about 3 hit non-hormone contraceptives, but we have no idea how they work. And so we're now trying to deconvolute that target, because if you're giving medication to people to either try and make them fertile or to make a contraceptive, they're not people who are suffering from some dreadful cancer or something that, you know, you're likely to take side effects and risk fairly moderately. These are entirely fit and healthy people that you have, you know, very little, if any, tolerance of risk at all. So it's really important that you understand what the drug target, what it's doing. So we think we understand what one of them is doing, but that's all locked down, so I can't possibly share that with you. But I think when you look at Joris's slide of all these different genes, actually you can see plenty of different pathways to do with ciliary motion or mitochondrial function or whatever, and things that are specific to sperm, which is quite exciting because the idea is then you're not going to have off-target effects for something else. So yes, we started off on a fertility drug discovery pathway. We pivoted because the grant funding came for contraception, but of course along the way there are side change that we can repurpose.
Hello. I work with a reasonable amount of men who have unobstructive azoospermia and with unsuccessful surgical sperm removal. And yet they hold hope that they will either conceive naturally or they will be able to improve their sperm enough to progress with IVF. And I'm just wondering what your thoughts are around men holding this hope.
Yeah, it's an important point, really. I mean, I think it's really important that when you're speaking to the patients, you give them very realistic ideas of the chances of success, because most individual IVF treatment cycles, treatment cycles, do not result in a baby. Factoring in uncertainty of sperm recovery, you're getting down to relatively small numbers. And we've been criticised before as a sector for selling false hope to patients about successes. So no, I think it's pretty unlikely. They'll ask you about scientific breakthroughs and discoveries. Well, you know, watch this space. But for the time being, nah. Sometimes they just want to get to the end of the journey, you know.
I'm quite interested in, Rod, the difference. You're talking about the testicular tissue being reimplanted and that being like a avenue of hope versus the spermatogonial stem cells. I'm quite interested in like organoid research, and potentially is there any sort of work being done to generate sort of testicular organoids that the spermatogonial stem cells could then sort of be used to understand sort of that pathway, if that makes any sense.
Yeah, so I guess that's the other option for fertility, is kind of in vitro generation of gametes or sperm, and you can do it with tissue in the animal studies. So you can take a bit of tissue from a prepubertal mouse and you can culture it, and you can get sperm generated that way just by putting bits of tissue that you could use. But people are looking at ways of generating organoids where you're combining all the cells that you need to combine to provide the right environment for those spermatogonial stem cells to develop towards sperm. You need all the factors to do it. You can't just put spermatogonial stem cells in a dish and hope that they will suddenly become sperm. So the tissue is, if you like, the tissue is already an intact environment. Everything's there as it should be. An organoid is an artificial environment that you're trying to generate. But people are working towards those. We don't have ones that are able to generate sperm in humans yet, but there's a lot of progress again on the animal side with that. So in vitro might be an option to generate those sperm in a dish. And the advantage of doing that over transplanting stuff back is for the patients who've got cancer, had cancer, that you'll have no risk of transplanting cancer back to the patients, which is one thing you have to think about for transplanting material back.
Is there any advice being given to men about who want to use them in order to reduce obesity, in order to become more fertile? Is there kind of a mixed possible effect there?
So yeah, is there a risk if you take
I think the simple answer is we don't really know. What we do know from men who've had bariatric surgery is if you drop your weight very quickly, it's not good for your sperm count in the short to medium term. So I've had a couple of quite interesting conversations as a clinician with couples embarking on IVF, in the very near future, to say, I wouldn't take this drug right now because I don't know how it will affect your sperm count, which seems really counterintuitive, but I think we just don't have the knowledge or the wherewithal. I think we understand a bit better that it shouldn't be taken during pregnancy by the female partner. We understand that because of the delay in gastric emptying, we often would ask them to stop it a month or two before they embark on IVF treatment, again, for the female side of it. But I think the concern is when you get into this kind of catabolic state where you're plummeting your weight very quickly, that it's that may have a negative impact on your sperm count. But it's a study that needs to happen, I think.
And I think you can see it would be very tempting for some people to pay for these weight loss drugs if it meant they were then eligible for NHS treatment, particularly in England, because losing weight takes a long time and you could be timed out in the age criteria bracket if you lose weight the hard way by dieting and exercise.
Yeah, I don't know other clinicians here, but certainly I see a healthy handful of people who have done exactly that. And who would blame them?
The de novo mutations that can cause or contribute to male infertility, as far as we know, are those somewhat random? So, for example, 100 years ago, they might have been the same thing. Or do we think there might be environmental insults that are causing them?
Right. Great question. So I'd love to give a lecture on this. So in general, basically when you reproduce your DNA, you can imagine 6 billion letters, you sometimes make a mistake. And this is what de novo mutations are. So they occur relatively random. There are certain positions where they can happen more often. What is interesting is that most of these mutations occur in the male germline, so during spermatogenesis, and the number of mutations that you get from your parents basically increases with the age at conception, right, of your parents. So if your parent, and this is again, 75% comes from the father, and an older father passes on more mutations to his children. So ladies here in the room thinking about reproduction, think about the man you choose. That's always my message. On the other hand, the risk of disease is not that much. But yeah, so you get about 100, they're all over the genome, and then it's, this is actually evolution, right? This is why we all look different. This is how it starts. De novo mutations are the start of how we change into new species and stuff. But unfortunately, these experiments more often go wrong than they go right. So they're an important cause of intellectual disability, all kind of rare diseases, and also infertility. But yeah, they occur relatively random. Yes, so they can. But if they affect the gene important in spermatogenesis, yeah, it's a big problem. And the testis is the tissue which has most genes expressed. So it's the brain and the testes, right? These are the two most important things.
Just another comment there. One of the genes that we discovered, what else do these genes do? One of them had tumor suppressor function. So in that way, you might see other ways in which we do see these observations of links between male health issues and fertility. It might be something to do with genetics affecting different sorts of pathways in health.
What do you think is going to show the most promise in the next 5 to 10 years? Apart from Sarah's research. Right, we'll go with Joris.
Starts with me. Okay, now, of course, I can only say, genome sequencing. Clearly, this is happening, right? I mean, I don't know how much you know about this, but in England, they do a newborn genome sequencing project. 100,000 children are being sequenced at birth, entire genome, right? So this is no longer science fiction. This is happening in routine care. And I think it'll be very important for fertility research to really understand what the causes are of infertility. So that's critical if you want to develop new treatments and move forward.
If you ask me back in 6 years, Sarah,
yep,
I promise you a baby.
Steady on. Not at my age.
Is something happening here?
Wow, that was an unusual answer, wasn't it? I think you better.
From a testicular tissue transplant. Sorry about that, everyone.
Phew. Those relieved. Yeah, I think it'll be in vitro gametogenesis. I think we will be able to make sperms and eggs in labs. I think we will be able to do that in 5 to 10 years.
And Sarah?
So I'm always a half full cup. And I'd like to think that we are going to ride this wave of increasing awareness of fertility, and it's going to be a much more open dialogue, that men are going to come to the part, and that actually, why is it, ladies and gentlemen, that we find it acceptable in medicine to treat the female for a male condition? So we need drug treatments, and I hope that in the next decade we might get somewhere closer.
Thank you very much.